Continuing with the development of pollen, I would like to talk to you about a very interesting phenomenon which is called as nemic phenomenon. So far you would have studied about the embryo sacs which are present in the ovules. Now I would like to tell you here that embryo sac are also present in the pollen grains. So there is a very interesting aspect, that is of occurrence of embryo sac like structures in pollen grains, and this was first of all observed by Nemec in 1898. He observed that there are 8 nuclei arranged in polygonum type of embryo sac in Hyacinthus orientalis. Now so far we only know that there is existence of embryo sacs in the ovules. Now the difference is that there could be a structure like embryo sac present in the pollen grain. It was hypothesized to be a result of 3 successive mitotic divisions in the vegetative nucleus of a pollen grain. So with 3 successive mitotic divisions, there are 8 nuclei which are formed, and these arrange themselves in the form of a regular embryo sac, which we refer to as polygonum type of embryo sac. Now later on this was confirmed by with 2 more scientists, De Mol in 1923 and Stowe in 1930. So it is like the microspores, they increase in size to form large sac-like bodies followed by 3 successive divisions of the nucleus. The 8 nuclei they arrange in polygonum type of embryo sac because of the divisions of the microspore nucleus. According to Stowe, it is a necrohormone which is responsible for causing an abnormal growth of the surviving pollen grains. You can see here in A, that is there is a small pollen grain which is a normal pollen grain. In figure B, there is an enlarged version of the same pollen grain, and there is formation of a pollen embryo sac after the single vegetative nucleus had undergone 3 mitotic divisions. In the C part of the figure, you can see a pollen tube entering the so-called egg cell, and in D it is the fertilized pollen embryo sac. So Nemec phenomenon is the formation of pollen embryo sacs, or we can say is the formation of embryo sacs in pollen grains. So this is very commonly observed in Hyacinthus orientalis. And then we talk about the germination of pollen grains. In this slide you can see that there are numerous pollen grains which can be seen putting forth small pollen tubes. What is pollen germination basically, and why is this germination of pollen so important in the developmental events? Now pollen germination is the first development event in fertilization. Now when we study plant reproductive biology, fertilization is the most important aspect of reproductive biology. In preparing for fertilization, the pollen has to put forth a pollen tube which will carry the male germ unit, which includes both the sperm cells and the vegetative nucleus. Now pollen germination involves hydration of pollen followed by emergence of tube and its growth. Now this pollen germination takes place when a partially dehydrated pollen grain, after dehiscence of the anther, it lands on a compatible stigma, and then it gets hydrated by absorption of locular fluid, or it absorbs the moisture which is present on the surface of the stigma. The growth of the pollen tube is confined to the tip region only, with the synthesis of pollen wall along the tip. Vegetative cell and generative cell also move towards the tip of the pollen tube. The wall components are synthesized by dictyosomes, RNA and proteins are synthesized during pollen germination and pollen tube growth. Now, For a pollen grain to germinate, there is requirement of certain conditions and certain compounds. Now, the first and the foremost is the relative humidity. Pollen are resting plant organs and are in a desiccated stage. They get activated by hydration. High humidity leads to higher germination, right? So it's important that there is good amount of humidity, there is good amount of moisture which is present on the surface of the stigma, so that the pollen can absorb water and then it can convert itself from a resting to an active state. Then come the carbohydrates. Carbohydrates are important both as a respiratory substrate, and they are also important in controlling the osmotic pressure of the pollen grain. Boron is another requirement for germination because it affects water relations and eventually it affects germination. It also helps in translocation of sugars and enhances the percentage germination. Boron also influences the biosynthesis of carbohydrates. Calcium is a very important component which is required for germination because it influences both the rate and the direction of the pollen tube. Individual pollen, they contain very little calcium, and this calcium generally diffuses out very rapidly whenever the pollen land on a medium, leaving a very low endogenous level of calcium. And the level of calcium, when it becomes very low, it is insufficient for pollen to germinate. So generally what happens is that when a large number of pollen, they germinate together on a semi-solid medium, this calcium, though it diffuses out, but if there are too many pollen grains on the surface, then this calcium is trapped between the pollen grains, and this trapped calcium, it furnishes the required level needed for germination, leading to germination. And now this phenomenon is called as the crowding effect or the population effect. Why it is called as the crowding or the population effect? Because it is the number or the crowd of the pollen grains which is important and which leads to the germination of pollen grains. Then there are flavanols. Pollen contain high levels of aromatic compounds which are derived from the tapetum of anther. Maize, petunia, they are deficient in flavanols. Since they are deficient in flavanols, they are self-sterile and they are unable to germinate. But if we apply some nanomolar concentrations of flavanols to stigma of these plants after pollination, this can induce germination. Then of course there are plant hormones, auxins and gibberellins. They promote germination in a very small way. There are enzymes which are very important in germination of pollen grains. These enzymes, they can cause elongation of pollen tube by rendering plasticity and elasticity to the tip region. Cellulase, pectinase and calase, these are present inherently in the pollen and are released immediately after placing the pollen in germination medium. So, if they are present in good amount, then probably they degrade the wall components and they help in the germination of pollen grains. The wall is, the pollen tube has the elasticity and can move further. The physical factors are also important in germination of pollen tubes. Pollen tube growth is enhanced by increase in temperature. Now 20 to 30 degrees temperature is the most optimum temperature for germination of pollen grains. In some incompatible pollinations, temperature can retard pollen tube growth. Now here you can see a scanning electron micrograph of pollen germinating in Tecomella. You can see that the pollen tube is labeled as PT. okay, the green one. These are the pollen tubes. On the right hand side we can see there are 3 pollen grains which are germinating, and you can see those thin elongated tubes here. Now once the pollen grain it germinates, it is important to know whether the pollen tube is able to carry the sperms and whether these sperms will be able to effect fertilization or not. There are two aspects of pollen grains. These are the pollen viability and the fertility. Pollen viability is the ability of pollen to germinate and to deliver male gametes to the embryo sac. On the other hand, pollen fertility is just the capacity of pollen to germinate. It is not important that if the pollen has germinated, it will be able to deliver the male gametes to the embryo sac. So, pollen fertility only refers to the ability or capacity to germinate. It may not affect the process of fertilization. Now, as you would have understood that the viability of pollen is very important, and this viability of pollen is affected by temperature and humidity both. If the temperature is low and the humidity is low, then the viability of pollen can be prolonged. Loss of viability is associated with irreversible loss of plasma membrane. Viability in some species can be as low as 30 minutes, or it can be extended up to several weeks. Now this delivery of male gametes by pollen is of the foremost importance. There are certain tests of pollen viability by means of which we can know whether the pollen are able to effect fertilization or the pollen will just germinate and the fertilization will not happen. The first and the foremost is the seed set or the ovule development, which we can see in the natural environments as well. Then there is pollen germination and pollen tube growth. Third is in vitro germination. There is tetrazolium test, fluorescein diacetate test, or it is also called as the FCR test, and nuclear magnetic resonance. Now starting with seed set or ovule development, this is the most reliable method, and this can be observed only once the pollination is over. In natural conditions, it can take a few days. Now after the pollination is accomplished, the fertilization has taken place, the ovary starts enlarging, and then there is seed formation. So we can observe that in few days after fertilization. Then there is pollen germination and pollen tube growth. Now pollen germination and pollen tube growth, in very few species you can observe the pistil under a microscope and observed. Mostly this is observed by fixing the pistils in acetic acid alcohol for 24 hours, and then they are later cleared in sodium hydroxide so that they become soft. So then these pistils are washed and mounted in decolorized aniline blue, observed under a fluorescence microscope. So under a fluorescence microscope we are able to see the pollen tubes. And there is one picture which I would like to show you here. Here you can see that there are pollen present on the stigma, and you can see pollen tubes traversing through the style, and eventually they reach the ovules. On the right hand side you can see in the second picture that there are small pollen grains, and you can see these rope like structures which are the pollen tubes. So pollen germination and pollen tube growth is the second method by which we can check the viability of the pollen grains. Then there is tetrazolium test. This is where we use 2,3,5-diphenyl tetrazolium chloride. It is also known as the TTC test. So TTC is prepared in sucrose. 12% sucrose is important to prevent the pollen from bursting, and the pH is adjusted to 5.8. Now what is done is that pollen are dispersed on a glass slide in a drop of TTC, and then they are immediately covered with a cover slip so that to exclude the oxygen, because otherwise the reagent will react with the oxygen. And the slides are incubated in dark for 30 minutes to up to 3 hours, and the temperature should be maintained around 35 to 40 degrees. Viable pollen grains, they turn red due to reduction of TTC to a red dye formazole. So this is a slide which shows these pollen grains which are stained with TTC. This arrowhead is showing a viable pollen which is bright red in color. Then there is a nuclear magnetic resonance test. Now this is one test which is non-destructive, and in this test we compare the levels of free and bound water within the pollen. If the pollen has lost the viability, then a considerable amount of water which earlier was in a bound state, it moves to a free state, decreasing the overall content of water. So with this you can evaluate the viability of pollen grains, and of course in lab we can use the in vitro germination method, whereas pollen are made to germinate in artificial media. Most of the times we use Brubaker's medium for observing the viability of the pollen grains in the laboratories. Fluorescein diacetate test is the most reliable test to check the viability of pollen grains, and this test depends on effectiveness of plasma membrane and presence of non-specific esterases in the pollen cytoplasm. In viable pollen grains, this FDR, this enters the pollen cytoplasm through the plasma membrane, and then it gets cleaved by the non-specific esterase enzyme which is present in the pollen cytoplasm. This releases a polar fluorescent molecule, fluorescein, and this fluorescein cannot move out of the plasma membrane, hence it gets accumulated in the cytoplasm. Species with ineffective membranes, they may give false reactions, right. Here you can see this FDA test, the bright fluorescing pollen grains are the ones which are viable. Now this is another picture of fluorescence micrographs of the pollen grains, and you can see that on the right hand side you can see there are two sperm cells which are fluorescing bright, and these have been stained with DAPI. Now, because we talk about plant breeding experiments, we talk about pollination experiments, and we also talk about germplasm conservation, it is important to store the pollen grains. Now, pollen storage is an important aspect of pollen biology. It is important for studies on various aspects of pollen biotechnology, pollen biology and pollen allergy, because then you can conduct different experiments if you have a good amount of pollen available with you. Preserving genetic resources and providing germplasm for international exchange is also one reason why pollen have to be stored. Pollen storage is important for overcoming crossability barriers imposed due to temporal and spatial isolation. Now, pollen storage, like I told you, is important for studying self-incompatibility, eliminating the need to grow pollen parents continuously for breeding programs, and implementing supplementary pollinations in order to sustain yield. Pollen can be stored under low temperatures. They are put in desiccators and then kept in refrigerators or deep freeze, and if we can store the pollen under conditions of low temperature and low relative humidity, the viability is extended. In mango, normal viability of pollen is up to 8 days, but at a low temperature of 4.5 to 9 degrees and low humidity, they can remain viable for 5 months. The other important method is to store pollen under ultra low temperatures, and this is done in liquid nitrogen, and viability is retained for a very long period as pollen undergo negligible metabolic changes at such low temperatures. Now, pollen can be stored by mixing with some diluents like Lycopodium spores, egg albumin, etc. They can be stored in organic solvents like acetone, benzene, ethanol, ether, chloroform and phenol. So, today we have learnt about the pollen germination and pollen storage, and I hope you have been able to understand the concepts of pollen development and pollen morphology. Thank you very much.